Good morning, children. Let's start with what we left yesterday. And we left at the applications of diffusion. I discussed with you all the factors affecting diffusion. That was temperature, density of the medium, distance, concentration gradient. And I think I was on the last topic, concentration gradient, how it affects the rate of diffusion. How concentration gradient affects the rate of diffusion. How concentration gradient affects the rate of diffusion. This is what I'm going to start. Now here, concentration gradient, as you know, I told you yesterday, like we have two setups, setup A and setup B. In setup A, we have two sides. In one, the concentration is different, and in another, the concentration is, suppose and here the concentration is 80 units. Again, here the concentration is 100 units in setup B, but here the concentration in the second site is 20 U. Now, if you see, the concentration is here and the difference of setup B is more here. That means the concentration gradient is higher here and it is lower here. So where the concentration in out of the two setups, where do you think the rate of diffusion will be faster, in setup A or setup B? Yes, who's going to tell me this answer? Where do you think the rate of diffusion will be faster, in setup A or setup B? That means from here to here, the diffusion will be faster, or from here to here, the diffusion will be faster. Where the diffusion will be faster, in setup A or in setup B? Yes, answer my question. Yes, Tadarani is saying setup B. Yes. Any other? Any other answer? Where the rate of diffusion will be faster? Yes. Anybody else who can answer this question? Yes, beta. Setup B, Siksha is saying yes. Any other? Where the rate of diffusion will be faster? Yes, any other student who can answer this question? Yes, Priya Yadav, Yashika. Amrita. I'm not getting any more response now. Okay, no problem. The rate of diffusion will be faster in setup B, where the difference is more, because there the concentration is very low, it will the rate of diffusion. Now, because the concentration gradient is higher here, concentration gradient is higher here, and concentration gradient is lower here. So where the concentration gradient is higher, there the rate of diffusion will be faster. Faster rate of diffusion. And in this setup, slower rate of diffusion. The rate of diffusion will be slower in this, whereas the rate of diffusion will be faster in that. Now, after this, we come to the next topic. So we have discussed all the factors affecting the rate of diffusion: temperature, density of the medium, distance, concentration gradient. So there were 4 factors which affect the rate of diffusion. We have discussed all 4. Now we move on to the applications of diffusion. Applications of where the diffusion is applied. Where can you see this process going on? Applications of diffusion. Now, if we see applications of diffusion, the first process where the diffusion is observed is exchange of respiratory gases. The first is exchange of respiratory gases. This takes place in a respiratory system in our lungs, and how this exchange takes place outside the lungs. Okay, outside the lungs, the concentration of oxygen is more, and inside it is less. So what happens in the alveoli, it is less. So from outside, the gas diffuses in. So if these are alveoli, one of the alveoli I have made, if this is alveoli, it has blood vessels around it. Here inside the alveoli, concentration of oxygen is less, and outside it, the concentration of oxygen is high O2 concentration. Now what happens in this case? From the higher, the oxygen diffuses in. So that is why oxygen diffuses in from outside. And same thing about CO2. Outside, the concentration of CO2 is less. CO2 is less outside. And inside, due to respiration, CO2 is high CO2. So what happens? It will diffuse out. The CO2 will go out. CO2 diffuses out because inside it is high. From inside, it will diffuse out. So this is the first example where the application of diffusion can be observed, and it is during the exchange of respiratory gases. The second process where this is observed is transpiration. The second process where this diffusion is observed is transpiration. Transpiration is loss of water in the form of water vapors. This is transpiration. When the water is lost in the form of water vapors, we call it transpiration. Now, how this water is lost? If this is stomata, if inside the stomata here, outside, outside, the concentration of water is less. It's very sunny, very dry. So outside the leaf, the concentration of water is less. And inside the plant, because plant is absorbing water through the roots. So inside the plant, the concentration of water is high. So what is going to happen? From inside, through the stomata, the water will escape in the form of water vapors. So this is the definition of transpiration which I have given you. So it is transpiration, the next process where this diffusion is applied. Third, where you can see the diffusion process is release of or diffusion of pheromones. Diffusion of pheromones. Pheromones are, yes, why? To attract opposite sex. Pheromones are special hormones which are released by the organisms during the breeding season. So this is what when the pheromones are released, so they reach the other partner by the process of diffusion. So this is the third where you can see the application of diffusion. The fourth process where you see the application of diffusion is proper distribution of substance between cells. Proper distribution of substance between cells. if there are two cells, adjoining cells, so the diffusion of substance from this cell to this cell, it will take place only by the process of diffusion. If one cell has less of that substance, another cell has more of that substance from the cell where it is more, it will diffuse into the cell where it is less. So this is the next application of diffusion where the process of diffusion can be seen. So here are 4 applications of diffusion. One is exchange of respiratory gases. Second is transpiration. Third is diffusion of pheromones to attract opposite sex. And then proper distribution of substance between the cells. So these are the 4 applications of diffusion. Now there is one more term which is known as facilitated diffusion. So here, I think this is clear to you all. Any point you want to discuss? You want to ask from this? Yes, is it clear, beta? Okay. So next we come to the facilitated diffusion. What is this facilitated? Facilitated diffusion is the passive transport of molecules across the membrane by some specific transmembrane integral proteins. So this is, we can say, passive transport. Diffusion is, of course, passive transport. So this is passive transport of molecules across the plasma membrane facilitated by or through specific transmembrane proteins, the intrinsic proteins. So that means diffusion is always a passive transport. No energy is required. So here also the diffusion takes place through the membrane. No ATP is utilized, but it doesn't take place normally like the diffusion. It takes place. It's not necessary that here, as I told you, for diffusion, the semi-permeable membrane is not required. But here in the facilitated diffusion, of course, the membrane is there. When it takes place through into a cell, when some substance is diffused into a cell, so there are specific proteins which are located in the membrane which helps the molecule to diffuse inside the cell. That means, although diffusion doesn't require any membrane, but in facilitated diffusion, if this is a cell and here the substance is, so it has to move in. How does it move? This membrane, plasma membrane, which is here, I have made it like this. This membrane has specific proteins here and there. These are known as integral proteins or intrinsic proteins. These intrinsic proteins which are here, these intrinsic proteins, they help these substances to move inside the cell. So this diffusion is facilitated by intrinsic proteins. This process of diffusion is made easy by the intrinsic proteins. And because this is diffusion, always the concentration of the substance will be higher here from where it is going and then to where it has to go. So always it will be higher outside and inside. This will be, the concentration of the substance will be less. Now, which substances are diffused by facilitated diffusion? It is fructose, glucose, nucleotides, etc. So these substances are diffused by facilitated diffusion. Now, if we see the membrane of RBC and liver cells, hepatocytes, the membrane of RBCs and hepatocytes. Hepatocytes means hepato means liver, cytes means cell, liver cells. There, through the membrane, the glucose is diffused. Glucose is diffused by facilitated diffusion. So this is one site where facilitated diffusion has been observed, and it is when the glucose diffuses inside the RBC from outside or into the liver cells from outside. So this is what about the facilitated diffusion. Now we come on to the next topic, which is active transport. Active transport. We have already discussed the properties of active transport, that active transport requires energy. It is energy dependent. The movement is against the concentration gradient. Always a semi-permeable membrane is present. So it requires ATP. Okay. Always semi-permeable membrane is present. Always a semi-permeable membrane is present. Okay. Requires ATP, always a semi-permeable membrane is present. Besides this, the movement is against the concentration gradient. Movement is against concentration gradient. Okay, and it gets affected by change in temperature. Change in temperature, okay, density, all these factors, they affect, all the factors which we discussed, they affect the active transport. So these are the general characteristics of active transport. Now, where is this active transport seen? The active transport, as I discussed before also, is seen in various pumps. So the first pump which we will discuss is sodium potassium pump. Na potassium pump. Where the word pump is used, pump means integral protein or intrinsic protein, intrinsic or tunnel protein. We discussed this intrinsic or tunnel protein when we were doing the structure of plasma membrane. I told you that in the phospholipid layer in between there are some proteins which are known as intrinsic proteins. They help in the functioning of sodium potassium pump. Now, what is the sodium potassium pump? Suppose we have this. Suppose we have the plasma membrane. This is our plasma membrane. And we have this tunnel protein or intrinsic protein which is located inside the plasma membrane. We have plasma membrane and this is the intrinsic protein. Intrinsic protein which is located in the plasma membrane. So we have this. Now what happens? The movement of sodium and potassium has to take place. Now how this movement takes place? So always, as it is active transport, it requires ATP. So ATP is utilized. How this ATP is utilized? The ATP is used, and this ATP, what it does, that when it gets broken down into ADP and P, inorganic phosphorus, with the release of energy. And this energy which is released by the hydrolysis of ATP, the energy which is released by the hydrolysis of ATP is required, is used in transporting the sodium from inside to outside. Sodium from inside is transported outside. So this is carried outside. 3 sodium at one time are transported outside, and in turn potassium, in turn 2 potassium from outside are taken inside the cell. This is the cell is like this. So this is inside of the cell, inside of the cell, and this is outside of the cell. Sodium at one go, at one time, when the ATP releases energy, 3 sodium are given out, and in turn 2 potassium are taken inside the cell. So this happens at the same time. Because of this continuous this process continuously, what happens? The sodium inside increases, outside, because sodium is transported outside, it increases. So I'll make another picture for you about that. This is another picture. This is the tunnel protein which I have made. So as a result, what happens? The sodium concentration outside increases. More of the sodium has increased outside. More of the sodium has increased outside. And inside, sodium is less now. Very, very little sodium inside, because 3 sodiums, they have gone out. So sodium outside increases and inside the sodium is less. Now everything tends to go from higher concentration to lower concentration. See, this is here the active transport has taken. Is it okay, children? Is it okay? Can you hear me now? Okay. Along with, I'll repeat this for you. I'll repeat this. See here, children. Active transport is taking place. The properties of active transport is that the properties of active transport is active transport requires ATP. It requires energy. It is always through a semi-permeable membrane. Active transport is always through a semi-permeable membrane, and it requires energy. And the movement is against the concentration gradient. That means even if in a cell something is less and outside, even if in a cell something is more and outside it is less, so it will move in. This is against the concentration gradient. So it takes place against the concentration gradient. That is why it requires energy. Then along with this, the movement is against concentration gradient. The example of this is, the first example is sodium potassium pump. Wherever we use the word pump, pump means intrinsic or tunnel protein. When we did the structure of plasma membrane, we made the structure like this, the plasma membrane. If you remember, I made this fluid mosaic model for you like this. Okay, the structure was made like this, phospholipid, phospholipid, and I made in between, I said that if there is some protein here which helps in the passage in carrying the substance from outside to inside or from inside to outside, this is known as integral protein or intrinsic protein or tunnel protein. So such proteins are present in the plasma membrane. Now if we consider this and this complete as plasma membrane, we have this tunnel protein here. We have this round one, the barrel shaped tunnel protein here. Now what happens when uh, this presence of this tunnel protein, it maintains the sodium potassium pump. How it maintains? When, see, as it is active transport, it requires energy. So ATP is is utilized, it's broken down into ADP and phosphorus with the release of energy. ATP is utilized, it's broken down into ADP and phosphorus with the release of energy. Now what happens? This energy is utilized to take out 3 sodium ions from inside the cell to outside. This energy is utilized to take out 3 sodium ions from inside the cell to outside. At the same time, 2 potassium from outside are brought in. This is this from where this energy comes to take out 3 sodium out and 2 potassium in. This energy comes from the hydrolysis or the breakdown of ATP to release ADPB with the release of energy. So now this happens. Now what happens? What is helping in this? The intrinsic protein, this intrinsic protein which I have marked here, this is helping because this transport is taking place through this intrinsic or tunnel protein. This is the intrinsic or tunnel protein which is there in the plasma membrane. Now what happens? When 3 sodium go out, the concentration of sodium outside the cell increases. See the concentration of sodium. This I made the plasma membrane. This is the tunnel protein, and this is sodium. The concentration of sodium outside increases. Now, when the concentration of sodium outside the cell increases, obviously what happens? From the higher concentration, it will come to the lower concentration. Okay, from here, the sodium will start moving inside the cell. So the sodium, when the sodium moves inside the cell, along with one sodium, one glucose also moves inside the cell. Along with one sodium, one glucose also moves inside the cell. Now you will say that this is, we cannot call it active transport, but this is here, this is a part of active transport. See here what is utilized? ATP is utilized to release energy, and because of this energy, 3 sodium are sent out and 2 potassium are brought in. Now what happens? Concentration of sodium, because 3 sodium are sent out, the concentration of sodium outside the cell increases, and inside the cell concentration of sodium is low. So what happens? Obviously, where the concentration of a molecule is high. Okay, beta, lower part is not visible. Is it okay? Is it okay? Okay. Now see the sodium from higher concentration moves towards the lower concentration. So sodium starts coming in. When sodium comes in, with one molecule of sodium, one glucose also comes in. Okay. Okay. So this is a part of active transport only. Rather, we give it another name. We call it primary active transport, and this secondary active transport. So this is known as primary active transport, and this is known as secondary active transport. One is known as primary active transport, another is known as secondary active transport. Now, one thing more. Here, if you see, there is exchange of 2 different ions. See, sodium going out, potassium coming in opposite direction. 2 different molecules are moving in opposite direction. So such transport is known as antipode. Antipode type of transport. When 2 molecules are moving in opposite direction, one is going out, another is coming in. So such transport we call as antipode. Now what happens here? Here if you see, both, whether it is glucose or it is sodium, both are moving in the same direction. Both are entering the cell, both are coming inside the cell. So such transport is known as sympode. Such transport is known as sympode. So this was sodium potassium pump. Is it clear to you or anything you want me to repeat? This is sodium potassium pump. Is it clear to you or anything you want me to repeat? Yes, beta. Clear, Tadarani is saying. Anybody else? Okay. Fine. So yes. Okay. So this was sodium potassium pump, which is which shows which shows active transport. Now I'll tell you one more pump where this active, yes, first I'll tell you the applications where this is applied, where you can see the sodium potassium pump. Sodium potassium pump can be observed, or this functions, or this is found in, or this is located in membrane of neurons. This is located in number one, membrane of neurons. Membrane of neurons. So if you see the neuron membrane is like this, we have negative charge outside and positive charge inside. Sorry, positive outside and negative inside. You must have done this nervous system with Lovepreet ma'am. So we have this electropositive here, electropositive, and electronegative here. The movement of impulse in this is based on the charges. The movement of impulse is based on the charges, and how this change of this charge takes place. It is through the membrane. It is through the membrane, and in this, the sodium potassium pump helps a lot. The second side where you can see this is the membrane of muscle fiber. Membrane of muscle fiber. There also change in charge is there for the conduction of the movement, for the conduction of contraction, or this, when a muscle contracts, that conduction of contraction, it is because of the change in the charge of the membrane of the muscle fiber. There also sodium potassium pump is located, and it helps in the change of the charge and for the conduction of the contraction of the muscle. Now second is this, now first we have discussed sodium pump. Now what I am going to discuss with you is sodium pump only. First we discuss sodium potassium pump. Now I'm going to discuss only sodium pump. There is another type of pump which is known as sodium pump. We have one sodium pump also, and same way we have one potassium pump also. So we have sodium pump, we have potassium pump. Now where is the sodium pump found? It is found in dendrites. The location of this is dendrites. Dendrites.